Hi, my name is Olga Lazareva, and today we'll talk about the limits of active model identification methods. First of all, let's discuss what are the active model identification methods. Here and further, we will refer to them as methods that take as an input experimental data, a phenotype information that can be disease subtype or survival time, and an interaction network. In contrast to using known pathways, active model identification methods have a potential to discover novel pathways by using a whole protein-protein interactome instead of known pathways. And there are many examples of how these methods provided valuable insights in various conditions such as hypertension, coronary heart disease, diabetes, and many others. But there is also plenty of factors that make researchers skeptical about protein-protein interaction networks. The degree of each protein or the number of interacting partners is largely affected by various sources of biases. So for instance, there is a technical bias that comes from the fact that bait proteins are going to have more interaction partners. Then there is a study bias when researchers tend to focus on the proteins that have already been proven to be interesting for the community, and therefore there is a strong correlation between the number of studies conducted on a protein and the degree of the protein. Finally, protein-protein interactions usually do not differentiate between tissues and conditions, and therefore often let us observe interactions that would never happen in the same time. Thus, we wanted to ask two important questions. Do networks actually make results more meaningful? And if so, why? So is it about node degree distribution or hub nodes, or actually the interactions themselves? To answer those questions, we have developed quite a comprehensive testing framework. So now I will walk you through it. And the first component is of course networks. We downloaded 5 commonly used networks: HPRD, APID, BioGrid, IID, and String. And also gene expression data for 5 different conditions. Then for each network, we have performed a set of alterations that allowed us to assess different qualities of these networks. To be more precise, we created 5 different network generators, each one to assess different properties of the original PPIs. The first generator is taken as an input the original network and then randomly switches edges such that the exact node degree is preserved. This way we preserve hub nodes and node degree distribution, but the edges are random. The next generator has a similar intuition, but instead of preserving the exact node degree, only the expected node degree was preserved. Then we try to shuffle gene IDs in a network such that node degree distribution preserves the same, but the hubs would be changed. The next permutation was based on just generating a random scale-free network and randomly assigning gene IDs to it. And the last generator was generating a random network where each edge had a uniform probability of existence. We can see now that the generators are ordered from the most conservative rewired to the least conservative uniform. Each of the generators assesses different qualities of networks, such as influence of exact node degree or expected node degree or the node degree distribution. Original and permuted networks, along with gene expression and phenotype information, are then fed into 8 popular methods. And each run is assessed with metrics that measure the functional association of produced gene set, and as well as the ability to explain the phenotype in question. The first metric that we used for the assessment was mean mutual information with respect to the known phenotype. The mutual information of 2 random variables is a measure of mutual dependence between the 2 variables. More specifically, it quantifies the amount of information that's obtained from one random variable through observing the other random variable. We computed mean mutual information between gene expression of the resulting gene set and the known phenotype or survival information. Then we also computed p-values to measure significance of association with disease-specific KEGG pathways. We also extracted disease genes from this GeneT network and computed overlap coefficient between the extracted disease models and these GeneT models. All in all, we performed over 10,000 runs to assess all possible combinations of networks, alterations, and gene expression. Here is the general overview of the results, separated by networks or methods. So on the x-axis, we see the original network and then all performed alterations. For a good network, we would expect that the scores on the original network are significantly better than on the random networks. And most of the times, we don't really see that, except for HPRD, so here. But this makes sense given that HPRD is the smallest network that was updated for the last time, I think, in 2010, so it probably has the most well-studied proteins. Similar pattern is observed with this GeneT overlap. Here we still observe that HPRD scores are a bit higher on the original network, but that's pretty much it. But it's, and it's also not the case anymore for phenotype-related metrics like mean mutual information with respect to the phenotype or with respect to the survival. So we had a similar expectation with respect to the methods. So a good method is supposed to produce much better results on the original PPI network than on the random one. To see if this is the case, we need to perform statistical analysis of the results to actually being able to tell whether the results here are higher than for random generators. So we made a per method comparison between generators and original networks. Higher is the point, more significant is the difference between the generator and the original network. But of course, this comparison only makes sense if the score of the original network was somewhat good to start with. So we are only interested in situations when the results were good on the original network and then became worse for generators. The size of the dot shows the proportion of good results for the method. So, for instance, we see that Domino here gave us significantly worse results on random networks, but the results on the original networks were good in 30% of cases. Overlap with this GeneT looks already worse, as methods either don't produce significantly worse results or do it only in 10% of the cases, like we see here with Diamond. And the situation is even worse for mean mutual information to the phenotype and with the survival. So the conclusion we got from this part is that methods mainly rely on the number of interactions a protein is involved in, but do not really benefit from the PPI themselves. But still, Domino, for instance, performed better than others, and it's important to understand why. First, we wanted to assess the topology of the produced methods and see how the genes are distributed in the original PPI network. We see that the Domino have the highest inter-gene distance in the produced models. This is probably due to the fact that Domino does not try to connect all gene models in one, and thus avoiding hub nodes. And that's also something that we observe on the right plot. Domino has one of the lowest correlation between the enrichment scores and the degree of its models. Cosine also, but unlike Domino's, cosine results were not too good on the original network. For active model identification methods in general, we see that there is a strong correlation between the mean degree of produced models and the enrichment scores. This means that a method that mostly relies on the hub nodes will produce same results as long as a hub node is in place. This we can also observe on the right plot, two generators, rewired and expected degrees that preserve hub nodes led to the same mean degree of solutions. And now we know that node degree itself is already enough to produce enriched pathways. We also tested so-called bias-aware methods, so hierarchical hotnet and netcore, but both of them were way too slow to include them in our main testing framework, and therefore we only ran them on the original versus rewired network and only for the smallest gene expression dataset. Neither of them, unfortunately, produced significantly worse results on the randomized networks. So, in conclusion, it's important to say that further algorithmic development is necessary. The encouraging results of Domino indicate that algorithmic improvement can actually overcome the node degree bias of PPI networks. Then different types of omics data and context-specific networks should be considered. For this study, we only evaluated the performance of the methods when run on gene expression data and PPI network, which is currently the most common case, but we expect that using different types of omics data and maybe context-specific networks can introduce new opportunities for active model identification methods. The next point is the quantitative measures of functional relevance of genes need to be used very carefully. So often we compare potential disease models to known disease-associated genes, but this approach is severely limited and biased by our current knowledge. And since the quantitative measures can be that biased, it's of course always a good idea to double check with domain experts. So I'd like to thank my co-authors for their help and support, as well as the chair of experimental bioinformatics, my professor Dr. Jan Baumbach, my supervisors Markus Liest and David Blumenthal. And of course, I would like to thank you for your attention. Thank you very much.